promoter called PR2.1, which was actually initially designed by Jeremy Nathans. He proved that this PR2.1 promoter, which is the red cone opsin promoter, human red cone opsin promoter, will in fact drive cone-specific expression in a transgenic animal. So we're using his promoter for many years now. 2.1 means the size, the length is 2.1 kilobases in length. Okay, first model that we studied was this GNAT2 model, a relatively rare form of achromatopsia, and the vector is the serotype 5 PR2.1 using the mouse GNAT2 cDNA, and that was published in 2007. So what I'm going to do is give you only a behavioral outcome for each of these animal models, not the ERGs, not the structure. Just have to assume it's there, or the referees wouldn't have accepted our publications. But the behavioral model for the mice, anyway, is this, as you know, this virtual rotating drum that has stripes that can rotate. And if they rotate to the right, the animal will move his head to the right if he can see the stripes, and he'll use only his left eye to do that. And you can then change the parameters of the stripes, so you can measure visual acuity by how wide the black and white stripes are. There'll be a point at which he can't see the stripes anymore. That's his threshold visual acuity. And of course, conversely, stripes go to the right, his head moves to the right, and he's using his right eye. I may have said right and left wrong before, but you could see what I was saying. Okay, so for GNAT2, I'm only going to show the one outcome here: visual acuity, untreated eyes, wild-type eyes in red, and the treated eye in blue, and you can see there's been an improvement to essentially wild-type visual acuity, mouse visual acuity anyway, with a very good p-value. Second model was for the A3 form of achromatopsia, one of the subunits of the cation channel. Again, serotype 5. We used a CBA, a general promoter here. We have, subsequent to this paper, we have used the PR2.1 as well, and it works just fine. And it's using, with the mouse, CNGA3 cDNA. Again, the optokinetic apparatus, visual acuity, again, a little bit different order here, but untreated eye, treated eye, that regains visual acuity back to basically wild type with a p-value of 0.05. Okay, we have another animal model for A3 achromatopsia. This is a recent one that's not published. It's really run through Eyal Banin in Israel at Hadassah University in Jerusalem. We've made the vector for this model. The vector is, again, AV5, PR2.1, in this case, human CNGA3, not sheep, not mouse, but human. And in this case, it's a behavioral test where the sheep has to navigate through this not so complicated maze, but sheep like other sheep. And so if you put the test animal over there, they like to go to their friends over here, if they can see it. And we'll see an untreated animal actually can't find his way through the maze, but a treated animal can. And here's the results. The untreated animal basically can't get through the maze, so there's really this is the amount of time it takes to get through the maze, but the treatment effect brings it right down to almost wild-type normal level behavior. So he can find his friends with one eye treated. Okay, the fourth model is a B3 achromatopsia model in dogs. There's also a mouse model, but I'm just showing you some of the results here. Again, serotype 5, PR2.1, and again, human CNAG B3, not A3. And that was published a couple of years ago, 3 years ago. Again, this is, John gave a nice introduction to this. This is the earlier maze that the Gus Aguirre and William Beltran constructed. I guess it was not William at that time, it was Andras Kamarmi and Gus Aguirre. But the dog has to find his way through this maze, and sort of that's what he's going to do if he can see his way with any efficiency at all. And the behavior is shown here: untreated eyes, treated eyes, wild type. So you can see that the treatment effect is really very profound, nearly again back to wild type behavior through this maze with a good p-value. Let's look now at a primate form of foveal deficiency, excellent color blindness. In this case, it's red color blindness, so it's protanopia, not a disease, not a blinding disease, a condition, but it mimics. It allows us to test the idea that we can deliver a cone gene to a primate fovea. Again, AAV5, PR2.1. In this case, the human red opsin promoter cDNA is what we're going to deliver. This is in collaboration with the Knights, formerly at University of Wisconsin-Milwaukee, now at University of Washington. And I can't go into a lot of detail on this, but the testing apparatus, they've actually, the Knights has trained the monkeys, the red colorblind monkeys, to select between 3 panels here. One of them has the test color, the others have some other color or neutral color, and the idea is they've trained them to choose the one panel that looks different from the other 2. So this is a treated animal. He's just touched with his nose the correct panel, so he gets a little reward. He's drinking a little grape juice. So the results are shown. This is the scales are complicated, and I don't need to go into it, but I just want to show you that a red colorblind monkey basically cannot see this wavelength or this wavelength of light. It's mixed with other wavelengths. It's really on a color spectrum. And this is squirrel monkey 1 and squirrel monkey 2. They both demonstrate that their protanopes, they don't see red well. They don't distinguish red from other colors, basically. This is pre-treatment in both of these animals with that vector, and this is post-treatment. Not quite back to baseline, which would be down about here, but you can see a big improvement in their behavioral ability to see red. They're now being able to pick out red at a much lower luminance than they were before. Okay, so actually in summary, I've shown you the 4 species and 4 genetic forms of foveal cone disease. Cones can be targeted by the right combination of AAV serotype and promoter, and can lead to behaviorally confirmed improvement in cone-mediated vision. Well, I had mentioned one other disease, blue cone monochromacy, and I haven't talked about it yet. Well, we're in the fairly early phases of it. We don't have behavioral studies. This is based on a rat BCM model that was discovered in China a few years ago. We're collaborating with them, and we have sent vector over there, and I've sent a surgeon as well to China to do these experiments, and they have subsequently done cone-mediated ERG analysis on these. I hope you can see this. This is a photopic flicker. Here is an example of a treated eye, nice flicker response, about 10 microvolts. And here is the untreated eye in the same animal, basically no response. The p-value is 0.008 over 6 treated eyes in 6 different animals. So we think that that there is going to be a behavioral improvement when we can get around to doing that. It's difficult from 7,000 miles away, but we're getting there. So basically we can maybe change the 4 species and 4 genetic forms to 5 species and 5 genetic forms, if you will. So basically, what's next? What are we going to do with this kind of information? Well, 3 things. Good news is that it looks like, according to the NIH, and this is sequester proof, I'm told, that we will get this, a gene therapy clinical trial initiated, the process for initiating a gene therapy clinical trial for B3 achromatopsia in April. So we'll see if that notice of award actually gets here. Secondly, since B3 and A3 achromatopsia patients have essentially identical clinical presentations, A3 patients will be discovered and characterized during the B3 natural history study. So A3 clinical trial will actually proceed in parallel with B3, and that will be separately funded through corporate support. So A3 and B3 achromatopsia are on track. And the BCM gene therapy clinical trial is under development, and that's funded with private support. So at least three of the major cone-specific foveal diseases, I think, have a future in the clinic, at least testing in the clinic. Thank you.
Okay, well, thank you very much for inviting me to come here and talk to you about our work today. So this is in the section called Gene Therapy via Viral Vectors, but we're not doing gene therapy and we're not using viral vectors, and in fact, this isn't optogenetics either. This is an alternative approach to restoring light sensitivity to blind retinas using chemistry, really. A group of molecules that we call photoswitch molecules. And so John Flannery already introduced you to this in retinitis pigmentosa and other neurodegenerative diseases of the retina. The photoreceptor layer gradually dies off, leaving the rest of the retina there, but sort of all dressed up with nowhere to go, not getting any light-dependent signaling anymore, and only over a long time, or there are changes to the inner retina. So the idea is that if you could somehow install artificially light sensitivity into these surviving neurons, you might be able to restore the perception of visual images by individuals suffering from these kind of disorders. So one way to do this is genetically, by putting optogenetic tools into these surviving neurons. But we've been using an alternative approach, which is developing small molecule photoswitches that can confer light sensitivity without gene therapy, and therefore could serve as sort of simple drugs for treating RP. So here's an example of the kind of molecule that I'm talking about. And these molecules have three parts. The central part is called azobenzene, which is photoisomerizable. So it changes shape with different wavelengths of light. On the one end of this azobenzene is a ligand that somehow interacts with voltage-gated channels and acts as a blocker in those channels. And then the other end of the molecule is a chemical group that either attaches these molecules covalently or just lets them linger around on the inside of ion channels. And so here's an example of one of these molecules at work. In this case, the blocker is tetraethylammonium, a classic blocker of potassium channels, and the other end of the molecule is sort of a greasy hydrophobic group. And so this molecule is membrane permeate. It accumulates in a cell, it integrates itself in the lumen of an ion channel, it blocks in one configuration, but when you photoisomerize it, it no longer blocks in the other configuration. And you can go back and forth between these cis and trans configuration between different wavelengths of light. So we've already published on the use of molecules like this for restoring light sensitivity to blind mice, in particular this RD1 strain of mice that essentially has retinitis pigmentosa. And so we use these mice months after birth when the photoreceptors are already gone. I'm sorry, one more thing about these molecules. This just shows the activity of one of these photoswitch molecules on a generic neuron growing in cell culture. And you can see when you put the molecule on these cells, as you block or unblock the certain subtypes of potassium channels in these neurons, you can induce action potential firing or cause that effect to disappear as you go back and forth between different wavelengths of light. And the photo effect happens quickly, reliably over many trials. There's no photo bleaching or anything like that of this molecule. And you can see the onset of the response is very, very fast. And you can do this in a more refined way by changing the equilibrium between the cis and the trans state. You can dial in how much excitability you want to have in a particular neuron. So of course, this then becomes an approach to restoring light sensitivity to the retina. So as I said, we use retina from blind RD1 mice where the photoreceptors are completely gone. We do recordings using a multi-electrode array with the ganglion cell side facing the electrodes in the array. And before adding our molecule, the retina is blind. And so you can see all these units, all these individual neurons, their activities are unaffected by changing the wavelength of light. And the average activity is flat as you go back and forth between different illumination conditions. But if you apply this AQ compound for just 15 minutes and then wash it away, the effect persists. Now you get all the ganglion cells responding robustly to light. Light onset causes a burst of firing of action potentials. This subsides a bit over time. Another wavelength of light returns the activity to where it started. So we have electrophysiological evidence, I'm not going to go through all the detail, that this molecule is working on many of the neurons that are surviving in the retina. And in addition, we have behavioral experiments showing that we can restore light-sensitive behavior. And for some reason, this didn't download perfectly well, so we're just going to do it the old-fashioned way. Here is a movie. Can we start from the beginning again? This is a mouse with no rods, no cones, and no melanopsin. If you just look at this eye, you will see that the flash of light after AAQ has been injected causes pupillary constriction. This is the same eye before injection of this photo switch, and basically the pupils are fixed and dilated, just the way they say on all the ER shows when a patient can't respond to light. So we're able to. There's many other behaviors that we've also been looking at, including light avoidance behaviors. Most recently, we have a fear conditioning assay, and we can show that mice can actually remember the pairing of the association between light and an aversive stimulus and respond accordingly over a long time after the photo switch injection. So this AAQ molecule that we've already published about has some problems. And the main problems are that it requires UV light, which is, of course, not going to be very useful in humans, partly because the lens and the cornea are just going to filter it out and it's never going to make it to the retina. It also requires really bright light, like standing on top of a mountain looking down in the snow at noon without your ski goggles on. And finally, this molecule dissipates rather quickly from the eye, requiring frequent resupply. So we've been in search of derivatives of AAQ that are more useful. And we now have a whole series of red-shifted molecules, shifted by at least 100 nanometer in their absorbance spectra. And the one that's the best is called DNAC, and it responds perfectly well to light in sort of the green part of the spectrum. In fact, you can use white light as well. And here is a recording, again, a multi-electrode array recording of a retina before DNAC application. This is in response to just flashing white versus black light. And here's the response after DNAC injection. So there's this really dramatic photosensitization of retinal ganglion cells. They're all responding with the same polarity. There's no on and off cells anymore, but they're responding nonetheless, and they're doing so to white light and even to green light and longer wavelength light. And if we look carefully at the intensity requirements for getting this response to occur, it turns out that it's actually quite good. I mean, it's quite reasonable levels of light. So the black graph here shows the intensity response relationship for AAQ, our original photo switch. And here is sort of the threshold light intensity that you need for getting a response. That, like I said, is very bright photopic conditions. And here is DNAC and a related compound called BNAC. They respond to about 100-fold dimmer intensity of light. And just for your reference, this is the response threshold of channel rhodopsin expressed in retinal ganglion cells, or halorhodopsin expressed in remnant cones. This comes from data from Botan Roska's lab. And you can see that the threshold for getting DNAC to respond, DNAC-treated retinas to respond to light, is about 10-fold dimmer light than for these optogenetic tools. So this is actually, other than melanopsin, this is really the best, most sensitive photosensitizer that we know about so far. So I mentioned that these compounds are drugs, essentially, so they will be metabolized. They'll wear off over time. But fortunately, and we don't exactly know why, DNAC lasts a lot longer than AAQ. So the red trace here is the decay of the AAQ effect in the eye, measured from different retinas extracted from mice. And it wears off within 12 to 24 hours after intravitreal injection. DNAC wears off over a week or so. The half-life is a couple of days. This molecule BNAC, which I also showed you in the last slide, can last for several weeks in the eye. It persists in the vitreous for a long time. And to extend this out even further, we're collaborating with Aaron Lavik, who's a bioengineer at Case Western Reserve, and we're encapsulating DNAC in polymer microspheres and nanospheres that can slowly release the compound over a period of many tens of days, months, So it's conceivable that molecules like this, with the right formulation, could install light sensitivity for a really prolonged period of time. So one of the really surprises that we've stumbled across in just the past couple of months is that it seems that the photosensitivity is specific to retinas that are exhibiting disease. This molecule only exerts its effect if the rods and cones are dead in the retina. So here's a recording from, again, a multi-electrode array, but in this case, we've put on a bunch of neurotransmitter antagonists to block all synaptic inputs to the retinal ganglion cells. And you can see that DNAC in this RD1 retina photosensitizes the ganglion cells themselves, and it does so very powerfully. If we do the same experiment with a wild-type retina, of course, the wild-type retina ordinarily responds to light, so we see a light response if we don't block the retinal circuitry. But if we block all the synapses and put on DNAC, we see no conferring of light responses to these cells. And we've compared now a bunch of retinas from strains of mice where the rods and cones are alive or the rods and cones are dead. The ones where they're alive include wild-type mice and mice that have gene knockouts that disrupt function in rods and cones and melanopsin cells, but don't kill off the cells. And here are two different RD strains of mice where the rods and cones really do die. This is RD1, which has a mutation in phosphodiesterase, and RD4, which has a mutation in transducin. And in both of these cases, the rods and cones die off within a month or so. And you can see that DNAC only makes things light sensitive if the rods and cones are dead. So this raises some really interesting questions. There must be some trophic influence of rods and cones over the ganglion cells that sort of introduces the target for DNAC, and we've sort of backed into this discovery that there's the target for this drug is something that is itself affected by the disease and in fact upregulated by the disease. So we're in search of what this ion channel type might be. So finally, DNAC wouldn't really be very useful if it were toxic. And, this requires a lot of work in larger animals, but at least at a first approximation, doing sort of anatomical studies, measuring the thicknesses of the outer and inner nuclear layers, measuring the density of retinal ganglion cells in the retina, we see no difference between sham-injected and DNAC-injected eyes. So it doesn't seem to be toxic, at least at first approximation. So just to summarize, these are some of the advantages of this DNAC molecule over our previous photo switches. It responds to visible light. Retina is treated with this compound. Respond to dim levels of light. The molecule persists in the eye for weeks, so obviating the need to constantly resupply it. It seems to affect diseased tissue, but not healthy tissue, which may also be useful in cases where only part of the retina is affected by disease, for example, macular degeneration. So far, we think it's safe and non-toxic, and it can drive visual behavior in vivo. And in looking forward to the potential clinical application of this, you could imagine, the sort of grand prize is restoring visual function in patients with end-stage photoreceptor degenerative disease. But because this is a small molecule where you can, theoretically adjust the dosing of it, you're not really overpowering the other systems in the eye by using a completely separate photosensor. You can imagine this being used to simply enhance low-level vision. Being a potassium channel blocker, it actually tends to amplify synaptic signaling. And also, just as a method for validating whether there's integrity in the visual system downstream from the retina, having a drug, a metabolizable drug that you can inject to just make sure that the brain is capable of receiving and interpreting signals from the retina, you can imagine using molecules like DNAC just to validate the functional integrity before undergoing other kinds of dramatic treatments, for example, implanting a retinal chip. So with that, I wanted to acknowledge the people in the lab that have done this work. There's been many people involved in this over the years. Most recently, Ivan Tochitsky and Alan Poliskina are two graduate students in the lab who have done all the multi-electrode array recording and behavior and other kinds of analysis with blind retinas. And then Dirk Trauner and Matt Banghart are the people who really invented these photo switch molecules that we've been using. So, and I didn't mention recent experiments with William Beltran at the University of Pennsylvania, who's doing experiments with blind dogs. We see the same sort of phenomenon, that DNAC can restore light sensitivity to retinal ganglion cells in blind dogs, but it doesn't seem to have any effect on the ganglion cells from normally sighted dogs. And all the different funding organizations that supported this work. So, thank you.
So the story started that a patient comes to see me. He works in a per se restaurant, and then he complains that he's bumping into things and get lots of cuts in his legs. And his friends thought that maybe he should get his eyes checked. Then eventually he found that this is almost a human equivalent for the LD1 mouse. It's a mutation in the phosphodiesterase alpha subunit, and one of them is a novel mutation. And the pictures of his eye, this is the optic nerve, and this is the right eye compared to a normal. You see the intraretinal pigment migration sparing the central macula. This is the right eye, and this is the left eye. And I'll highlight the purpose of the mouse project, that when most patients, when they first come to see us in the clinic, they already in mid or late stage of the disease already. Versus like in some of the previous, at least mouse models that I have done, they come before the onset of the disease. So they will be the purpose of the project. So this is his another patient of phosphodiesterase mutation, so almost equivalent of the LD1 mouse. This patient also get a lot of epiretinal fibrosis after membrane peeling. You can see the macula, and it improved the central vision by about two lines. So this is a phosphodiesterase gene that so for gene therapy, some of the early work done by Derba and also done by Bill, that you can just increase the phosphodiesterase with a virus, and we have done both lentivirus and also done with AAV more recently. And so you increase the function of phosphodiesterase, and now break down 3 parts of the talk and show you the story that we have a mouse model that now we can now treat to study the mid phase or late stage of the disease instead of beginning before the onset of the disease. And then the story for this new mouse model teach us that actually you need to save enough rods in order to prevent the cones from dying. The whole retina may reach the stage of no return at some point, so you need, unless you cross this threshold. And then in a combination of gene therapy, maybe you need some, putting back the effective gene phosphodiesterase. You may also need to do something else and to prevent crossing this stage of no return. And I'll have some suggestion, maybe manipulation of the cell survival mTOR pathway. So just put the gene back with the CDN using aldopsin promoter. This is a injection at this point with tag with a red fluorescent protein. You can even see the needle track here. And this was done before the mice get the degeneration, so you can envision gene therapy in a neonate or situation. Then one single injection essentially have last more than almost a year now. So this is the one is a treated eye, and the other is a fellow eye. So almost a year, one single injection. But we also found that if you treat treated early before the onset of degeneration, this is treating a later stage, a mid-phase degeneration, that as time goes, you treat them at early stage of degeneration, the efficacy is always better than you treat them at the late stage or mid phase of the disease. And because we're different students and postdocs doing injection, our surgery probably is not as skillful as the surgery done in Bill and done with Deborah, we decided not using a systemic drug to turn on the gene instead of using every time using viruses. So we designed a phosphodiesterase allele that have a stop cassette. In the presence of a drug, this allele will turn on, and so this will block the expression of this phosphodiesterase, and the presence of a drug-inducible recombinase will pop out this cassette, and then this will be on. So we can now give a systemic drug instead of doing a virus every time and take away the variability of surgery. So this is just shown in here. Maybe you can play the video. So these are the drug, you can give it systemically, and we'll pop out the stop cassette. And then the phosphodiesterase activity will be like the blue protein. So this is shown in a control mouse. Is autofluorescent imaging. This is a calibration bar that we can standardize the amount of autofluorescent in each mouse. This is a heterozygous. There's no essentially a normal control, positive control, and this is a disease mouse with retinitis pigmentosa with no treatment at a early stage and a little bit at a later stage. So you can see that vessel become attenuated, you start getting spots. And then in the, if you treat the early rescue majority of the rods at the early stage treatment, you can get pretty much normal compared to control. If you start treating at a later stage, if you do not treat more than half of the rods, then you get the treatment is not as efficacious, only slightly better than the negative control. This is looking at histology. This is again the control. You see the rows of nuclei. This is a positive control. This is a normal, pretty much normal mouse, and this is at this stage a single row of nuclei. If you treat the mice you cover saving a lot in the early stage of the disease, they can get pretty much closer to normal. You start treating them at a late stage of disease that is not as efficacious. He's looking at the ERG responses. For example, look at this box. If you treat them at a late stage of disease, there's not much different from the negative control. You treat them at an early stage of disease, then you can get back almost close to normal. And then this is, I will need a lot of feedback to interpret this picture. This is the peanut stain that Janet talked about on the cones. So this is a positive control. This is a normal mouse, and this is the mutant mouse at the late stage. With then you get these cones a funny-looking shape. If you treat them at a late stage, or you rescue not enough rods, then you get all these funny-looking cone cells. If you treat them at an early stage, then you get a mixture of funny-looking cells and normal-looking cones. And this is the same case as in patient. This patient C2020. You can see it's a high-density central autofluorescent ring. This is a patient with a phosphodiesterase mutation. By OCT, looking at the central fovea region, but OCT looks pretty normal. And then in adaptive optics, looking at the cone cells, so this is close to the central fovea region. In a normal patient, you can see the cones as you go later, farther away from the fovea. In a normal patient, the cones get less and less. And at this stage, even though this patient C2020, and you see at almost normal by OCT, the cones already looks funny already. So almost at this stage looks like the mouse picture on the peanut stain. So this would suggest that you probably need to there's a threshold number of rods you need to rescue before the cones start looking funny looking, which I don't know how to describe these cones other than they look funny, or some kind of dysmorphic cone. So even putting the cDNA, putting the gene back may not be sufficient because most of our patients come to clinic, they do not come at early stage of disease, maybe they come at mid and late stage of the disease. So there's a region pretty important that I'm going to highlight the inner segment, outer segment junction, or the ellipsoid inner segment. So you do OCT staining, there's high density